So good morning. We are here this morning with my colleague Laurent Groc to announce the publication of a paper dealing with the management of the severity of L-DOPA-induced dyskinesia in Parkinson's disease. The paper is soon to be published in Journal of Clinical Investigation, and it is a very deep and strong collaboration between two laboratories of neuroscience in Bordeaux, the Institute for Interdisciplinary Neuroscience, represented by my colleague Laurent Groc, and the Institute for Neurodegenerative Disease, represented by myself. What we've done in this study is that we have built up upon our former discoveries stating that the dyskinesia might be related, at least in part, by the anchoring of the dopamine D1 receptor at membrane in the brain of PD patient, of non-human primate-made Parkinsonian, and also of rodent-made Parkinsonian. So building up upon this series of discoveries, we have now tried to release the dopamine D1 receptor by interfering with its interaction, which was not very well known, with PSD95, one of the main components of the postsynaptic density. By doing so, using either downregulation of the expression of PSD95 using gene therapy, or by using peptides specific of the interaction between PSD95 and the D1 receptor, we've been able to improve the severity of L-DOPA-induced dyskinesia, first in rat models and then in non-human primate models. The problem was, were we actually releasing the movement of the dopamine D1 receptor?
Okay, so as Erwan just explained, this work has been really based on a strong collaboration between two institutes. And what we did in our laboratory is to uncover the cellular pathways and that were dealing with the interplay between PSD95 and dopamine D1 receptor. And for that, Amandine Berthet from Erwan's lab came to our lab and play a bit with the single nanoparticle approaches that we developed over the last decades. And this allowed to follow a single receptor on the living neurons and to understand what regulates the distribution of these receptors. And in this study, we, for the first time, showed that we can play with the trafficking of a dopamine D1 receptor in the striatal neurons. And by doing so, we could interfere with the distribution of the receptor and their signaling. That in some pathological case, such as in the Parkinson's disease, of course led to some disorders. So this work has been clearly one of the first demonstration that interfering with the dynamic of membrane receptor at the surface of a living neuron is a key process to interfere with the signaling of these receptors and to the abnormal signalization that can occur in some pathological disorders. So this is clearly the first time that we show that, and I guess it won't be the last part of the ongoing and very exciting collaboration between our two institutes.